Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about our next part of X-ray fluorescence spectroscopy. So in the previous lecture we have discussed about the detail of X-ray fluorescence spectroscopy, that is the introduction, their principle, their advantages, disadvantages and the parts. So today we will going to study about out the two main X-ray fluorescence methodology. So basically there are two methodology of X-ray fluorescence spectroscopy. The first one is energy dispersive and second is wavelength dispersive. So let us discuss each methodology in detail. First we will going to cover the energy dispersive X-ray fluorescence spectroscopy, then we will going to discuss the wavelength one. So what do you understand by energy dispersive X-ray fluorescence spectroscopy? So energy dispersive spectroscopy, it is basically it is used it makes the use of the X-ray spectrums which emitted by a solid sample and they further bombarded with a focused beam of electron to obtain a localized chemical analysis. Okay, so now let us discuss this in detail. So the energy dispersive spectroscopy, it is also known as EDS technique. It is basically used or mostly used for the qualitative as well as quantitative analysis of material, but it is also capable of providing a semi quantitative result as well. Okay, so it is not always the case that it will only used for the analysis of qualitative of analysis of the material. No, for the both the one qualitative as well as the quantitative. Okay, so what does it mean? Let us understand with the help of a definition. So energy dispersion detection system directly measure the different energies of the X-rays from the samples. Okay, energies emitted of the emitted X-ray from the sample. So by counting and plotting the relative number of X-ray at each energy and XRF spectrum is generated. So the principle of energy dispersion, that is ED detector, it is based on the generation of electrons hole pair in a semiconductor material often known as silicon. So in this the incident ray of energy EX is observed by the detector material and will cause one or more electron hole pair to form. The energy EHP do not do to do this is fixed for the particular material. So the X-ray will form as many electron hole as its energy allow. The number of electron hole pore pair is given by EX slash or divided by EHP. Okay, so now let us understand what does it mean in detail. Okay, now let us understand this with the help of the principle. Okay, so if we talk about the this energy dispersion X-ray fluorescence spectroscopy, so in this the incident beam, what they do, they may excite the electron in the inner shell. So let's see that this is an electron, this is an orbit. Okay, so here it is a nucleus. Okay, here it is a nucleus. I'm just using different pens. Okay, so what does it means? So when the incident light, let's say this is an incident light, so when an incident beam or light, what they do, when they fall, they may excite the electrons. So here these are different shells. So what they do, they will excite the electron. Here the electrons get excited, they may excite the electrons in the inner shell. Okay, inner shell, the exiting it from the shell while creating a electron hole where the electron was. Like simple सी बात है, अगर मैं हिंदी में इसको समझाऊं तो होता क्या है कि जब भी एक बीम ऑफ लाइट पड़ी, ठीक है, इस शेल पे पड़ी तो क्या होगा, और बेसिकली जब इनर शेल पे पड़ेगी तो जो इनर शेल पे इलेक्ट्रॉन प्रेजेंट है वो एक्साइट हो जाएगा. जैसे ही वो एक्साइट होगा तो वो बाहर आने की कोशिश करेगा. इट विल फोर्स इटसेल्फ टू कम आउट. इट विल इजेक्ट फ्रॉम द शेल. आफ्टर इजेक्टिंग फ्रॉम द शेल, जैसे ही वो शेल से बाहर आएगा तो यहाँ जो उसकी प्लेस होगी, लेट्स से ये वाली प्लेस होगी, वो क्या हो जाएगी, वो खाली हो जाएगी और वो वहाँ पर एक इलेक्ट्रॉन होल क्रिएट हो जाएगा. Okay, in Hindi this is the whole phenomena. So what is the phenomena? When the incident beam may excite an electron that is present in the inner shell, it will eject out from the shell while creating an electron hole where the electron was originally associated. So the electron from an outer higher energy shell then fills the hole. So here means the adjacent shell, let's say this is a electron in the other shell, तो यहाँ पर जो होल है इसको कौन भरेगा, ये इलेक्ट्रॉन भरेगा जिसके पास हाई एनर्जी shell then fills the hole. The difference in the energy between the higher energy shell and the lower energy shell may be released in the form of X-ray. Okay, so the number of energy of X-ray emitted from the specimen can be measured by the energy dispersive spectrometer. So this is the whole phenomena of energy dispersive spectrometer. Again I am just repeating, the incident beam, it may excite an electron in the inner shell. The inner shell electron, they eject out from the shell while creating a electron hole where the electron was originally present. Okay, so when the hole was created, okay, after the electron moves out, so the electron from the outer shell, the higher energy shell, then fills the hole, and the difference in the energy between the higher energy shell and the lower energy shell may be released in the form of the X-ray. Okay, so the number and energy of the X-ray emitted from the specimen can be measured by energy dispersion spectrometer. Okay, so this is the whole phenomena. Okay, now let us understand what are the different instruments that are used in energy dispersion spectroscopy. If you talk about the energy dispersion spectroscopy instruments, so the first one is the electron beam source. The second one is X-ray detector. Third is analyzer, and last is your pulse processor. Okay, so what does this electron beam source function is? So if you talk about the functions of the first one, that is the electron beam source. So the function of electron beam source is as like so basically what is the basic use of this? So it is used to focus the beam lengths are used along with the aperture. Okay, so the energy of the electron beam has to be very carefully selected to overcome the compromise between this resolution requirement and the production of X-ray. So produced X-ray are detected by the two crystal spectrophotometer. Then the second one is your X-ray detector. So the X-ray detector, it counts ounce, which is the abundance of emitted X-ray, versus X-ray energies that are measured by the EDS detector. So because since the name suggests detector, it is a solid state device which is used on based on the lithium drifted silicon. Okay, so the most important part of here is the detector. So basically if we talk about the detector, so detector, they are basically of, if we talk about the part of the detector, okay, so part of detectors that are used in energy dispersion spectroscopy, the first one is your collimator assembly. Okay, now the second one is your electron trap. Electron trap. Okay, so what is the function of the first type of detector? So the first one is the collimator assembly. So the purpose of the collimator is to provide a restrictive aperture through the X-rays should pass in order to make a way to a detector. So here the collimator ensure that only X-rays from the region being excited by the beam of electrons, they are get detected. Then comes the electron trap. So the function of the electron trap, or you can say the purpose of the electron trap, is to guarantee that only X-rays should enter the detector. Means no other rays should not enter the detector. Only which type of rays should enter the detector? Only the X-ray should enter the detector. So it has the pair of permanent magnets to strongly deflects the passing electrons. Okay, so this is the basic purpose behind it. Now the most important part of this segment is the application. So if we talk about the applications, so I'm just writing the applications. If we talk about the application of electron dispersion fluorescence X-ray spectroscopy, so it basic the first application is the study of microorganism. This technique is basically used for the study of microorganisms like bacteria, viruses, pathogens, and have made of the treatment diseases very effective. Okay, the second application, it is used to study the topography of element used in the industry. So those elements which are used in the industries, so they are basically, this technique is used to study the topography of the element. Then the third use is, or application is, used to analyze the lead based paint. Okay, third your last one is the agriculture traces metal analysis in the soil and other irrigation products. So these are further applications that have a you can say that where the energy dispersion X-ray spectroscopy is used. So that brings to the end of the first methodology. Now let's move to the second methodology, that is the wavelength dispersion spectroscopy. So the wavelength and dispersion detection system physically separate the X-ray according to their wavelength. So the basic principle or aim behind this methodology is to separate the X-ray according to their wavelength. Means according to their wavelength, the X-rays get separated. So this point should be an important one. According to their wavelength, okay, they separate the X-ray. Now the second says the X-ray, they are directed to a crystal. Okay, so this crystal, what does this crystal do? They diffracts the X-ray in different direction according to their wavelengths. Means after the X-ray, they are passes through a crystal. What the crystal do, they diffracts, means they diffracts the x-ray in different direction according to their wavelength, or you can say energies. So on consequential system of detector is placed at the fixed position, the crystal is rotated so that the different wavelengths are picked up by the detector. Okay, now comes the thing that the XRX spectrum is built up to the point by point in stimulus system, sorry, simultaneous system. The number of crystal or detector are used, so the range of element can be detected simultaneously. Okay, so this is the principle behind the wavelength dispersion spectroscopy. So if again, what does a wavelength spectroscopy, again I am just telling you in a detail, what do you understand by the wavelength spectroscopy. So wavelength spectroscopy says, so this type of technique or method, or you can say methodology, the basic funda behind is to separate the x-ray according to their wavelength. Then when the x-ray they passes through any crystal, so the crystal they diffracts the x-ray in different directions according to their wavelengths. Okay, so on a subsequential system, so the detector is placed at a very fixed position. So this is very important, students, and the detector should be placed in a very fixed position. So position of the detector should not be changed. So and the crystal crystal is rotated, so and the different wavelengths are picked up by the detector. Okay, so the when the crystal is rotated like this, see if this is a detector, this is a detector and crystal is placed here something, this is a crystal, so crystal is rotated around this detector, so that detector could detect the presence of the x-rays of different wavelength, or you can say different energies. Okay, so this is the principle behind the wavelength spectroscopy. Now moving to the next, that is the principle difference between the ED and the wavelength dispersion technique. So we have already covered the two methodology. So now comes what is the difference or principal difference between the ED and wavelength techniques. So basically that technique they lies in the achievable energy spectral resolution. So the wavelength dispersion X-ray fluorescence spectroscopy system can routinely routinely provides the working resolution between 5 electron volt to 20 electron volt depending upon their setup. Whereas if we talk about the energy one, so they provide the resolution that ranges from 150 to 300 EV or more depending upon the type of detector used. So this is the first principal difference between the two techniques. The first one is the VD means wavelength, and second one is the energy. So wavelength it provide the working resolution between 5 electron volt to 20, whereas the energy gives a higher resolution, that is 150 EV to the 300 EV or more. So depending upon the type of detector they are using. So the higher resolution of a wavelength dispersion X-ray fluorescence spectroscopy. So because since I have just told you the reverse one, so wavelength here it is providing the higher resolution, so it is it provides the advantages in reduced spectral overlap, so that the complex sample can be accurately characterized. Since the wavelength one is giving the more higher resolution, so what they do, it reduces or it it minimizes the spectral overlap. When they minimizes or reduce the spectral overlap, so the complex sample means the samples that are very complex in nature, they can be accurately characterized. So in addition, with the higher resolution, backgrounds are reduced, providing improved detection limit and sensitivity. Okay. Now moving to the next, that is how does XRF instrumentation work. So this one is again a very important one slide, that is how the X-ray instrumentation work. Okay, so the first one is X-ray generation. So the XRF instrument contain X-ray tube that produces X-ray. So how does a X-ray fluorescence spectroscopy work, or means what are the different components that are there in the instrumentation? So if we talk about the first one, then it is X-ray generation. How the X-ray are generated? So X-ray instrumentation, it contains a X-ray tube. So this X-ray tube, what they do, they produces the X-ray. After the production of X-ray, in the X-ray tube, electrons, they are accelerated and further they collide with the target material, usually a metal such as RH and Ag, to produce the high energy X-rays. Okay, now comes the sample irradiation. Now the second one is the sample irradiation. So the generation of X-ray are directed towards the towards the sample being analyzed. So when the X-ray hit the sample, whenever the X-ray is hitting the samples, they interact with the atom in the sample. After the interaction of the X-rays with the sample, they emit the characteristics X-ray which are unique to each element. Now moving to the next, that is the detection. Okay, so the instrument, it contains a detector that can identify and measure the energies of the characteristics X-ray emitted by the sample. So the intensity of these X-ray is proportional to the concentration of corresponding element in the sample. Now comes the analysis. So the detected X-ray energy spectra are analyzed by the instrument software. The software computers the characteristics of X-ray energy to a known database X-rays of for different elements. So based on this comparison, the software determines the environmental composition and concentration. So for this, for the analysis of any any sample, so the elemental composition and the concentration of the sample play a very important role. So because of this two, the software determines the environmental composition and the concentration in the sample. So the user's software, it represent all the elements and concentration in a table, in the table form they where the user represent, and that is very easy to read, and if required to transmit it to an external computer or local servers for further analysis or backup. So this one is very important, that the user software, it presents all the data, means it presents all the elements and their concentration in the form of a table. That table make it easy for the read, and for further backup point also they are very important. Okay, so now brings to the strength and limitation, or you can say advantages and disadvantages of X-ray fluorescence spectroscopy. So if you talk about the strength, if you talk about the advantages of X-ray fluorescence spectroscopy, so X-ray fluorescence spectroscopy, it is particularly well suited for the investigation that involves bulk chemical analysis, where you want to analyze the bulk chemical analysis of major elements like different, like manganese, magnesium, calcium, potassium. Okay, so in the rock and sediment, so basically it is used in the analysis of bulk analysis of major elements that are present in rocks or in sediments. Now comes the bulk chemical analysis of the trace elements. So where you want to again you want to analyze the bulk chemical analysis of the traces elements that for example that are more than 1 ppm. So basically there also this type of fluorescence spectroscopy play a very major role. Now comes to the limitation. So in theories the XRF has the ability to detect the X-ray emission from vertically all elements. So depending upon the wavelength and intensity of the um, incident or X-ray. However, in practice, most commercial available instruments are very limited in their ability to perceively and accurately measure the abundance of elements. So this is one of the major limitation or disadvantages with X-rays spectroscopy. So it also cannot distinguish between various type of isotopes that of elements. So if you want to distinguish the variation among the isotopes so of the element, so this could not analyze. So these analysis are routinely done with the other instruments. So these are certain disadvantages. Now moving to the end of the lecture, that is the application part. So X-ray fluorescence is used in a wide range of applications that include research in igneous and sedimentary and metamorphic petrology. Okay, so whenever you want to survey soil, so this application is widely used there. In mining, whenever you want to measure the grade of an ore, so this type of technique have a huge applications there. Again, in the cement production, in ceramic and glass manufacturing, so you can see that up to every industry it has its application, whether it's a cement production, whether it's a ceramic or a glass manufacturing, or whether it is a quality control of meteorology, environmental studies, even in the petroleum industries, field analysis, or in geographical and environmental studies. So it has a huge applications there. Apart from this, it has a huge application in microbiology, where you want to detect different types of microorganism. So it has a very huge application criterias where XR fluorescence spectroscopy is used. So now if we talk about the research and development, how X-ray fluorescence spectroscopy helps or you can say have an application in research and development. So X-ray fluorescence spectroscopy, it play a very pivotal role in material research and development. Like material sciences means researchers studies the properties of materials, and it creates a new compound, that is catalyzes or coating the material by precisely and accurately analyzing the elemental composition. Okay, in semiconductor industries it has a huge advantages, like applications, or you can say X-ray contribute a developing of cutting edges of semiconductor by ensuring a purity and composition of material used in a chip manufacturing. Art and archaeology, that we have already discussed, so it has a huge application in every field. So in examination also they can ask you, means how X-ray fluorescence spectroscopy has have different applications in research and development. So you can tell all these 4 points. Now moving to the next, that is the end of the lecture. But before ending the lecture, let us discuss some important questions. So for the important questions, the methodology part is very important. The two methodology, what should be the in examination or in interviews they can ask you about the difference in the principal difference between the two, that is energy dispersion and wavelength dispersion, X-ray fluorescence spectroscopy. So you must be knowing that. And the instrumentation part, they can ask you about the advantages and disadvantages. But yes, the most important one is the methodology and the application one. So that brings to the end of the lecture. I hope you have understood the lecture. If you have any doubt, any queries related to the lecture, you can simply leave your queries in the comment section below. In the next lecture I'll be answering all the queries. And yes, student, don't forget to subscribe ITLS Academy. You can you can subscribe ITLS Academy and get updated. Thank you for watching. Please do like, share and subscribe. Thank you.